Have you ever wondered how building cities affects plants and animals? Unfortunately, scientists have documented great losses of plants and animals due to the effects of urbanization. You see, when we modify landscapes to fit our human needs, we destroy habitats and create barriers such as busy roads that animals and plants cannot travel across. This can cause populations to become genetically isolated from one another and affect the ability of species to persist. Additionally, cities experience increased temperatures that can alter organisms' physiological development and increase the spread of diseases. Interestingly, bees, the pollinators we rely on for crop production and pollination of our wildflowers and gardens, may actually benefit from low levels of human disturbance. For example, here in the Raleigh-Durham area, bee communities in single-family home neighborhoods are more abundant and diverse than those in more natural settings such as state parks. One reason for this increase in bees could be that home gardens tend to have many showy flowers that attract bees to settle in these residential neighborhoods. However, many of these species in home gardens have been bred to have big, beautiful petals, perhaps at the cost of nectar and pollen, the food resources that bees need to survive and raise young. If bees perceive the increase in these flowers as an increase in resources, they may disperse to residential neighborhoods and not reproduce there, making residential neighborhoods a sink or an ecological trap for bees. The goal of my dissertation is to determine whether bees are successful in urban residential habitats and why. This will help us understand how to best conserve bees in an increasingly urban world. This past summer, I got bumblebee workers from 16 sites across the Raleigh-Durham area and non-lethally removed a portion of one leg per bee. These genetic samples will enable me to map gene flow within the urban habitat matrix and to identify potential source sink dynamics. To estimate the reproductive success of bees, I will place experimental bumblebee colonies in residential neighborhoods and public open spaces such as state parks, which will serve as a baseline for bee success. I will monitor these bee colonies in the field to measure various fitness correlates such as how fast workers are produced, as well as how many reproductive individuals the colony produces at the end of the season. I will also monitor worker foraging rates and sample the pollen they carry. Using a reference collection of pollen from each plant in my field sites, I will analyze worker pollen loads under a microscope. If pollen composition correlates with colony fitness measures, I will test pollen quality to understand how we can increase resource availability for bees in urban habitats. The broader impacts of this research include identifying conservation habitats for the native bees that pollinate our flowers and offer economic insurance for agriculture in the midst of honeybee collapses. Ultimately, I aim to inform land managers, urban planners, and policymakers on how we can conserve native bee diversity here in our urban neighborhoods.